Good afternoon, everybody. Welcome. My name is Alicia Lehrer, and I'm the Executive Director of the Woonasquatucket River Watershed Council. We're going to get started in just a minute, but I wanted to take a few moments to allow a couple more people to join. So just hang tight. We'll be right with you. Good afternoon. If you just arrived, we're just giving it a couple minutes before we get going, probably just one more minute, and then we're going to have our exciting lunch and learn session. So we'll be right with you. Okay, I think we're ready to get started. Good afternoon, everybody. My name is Alicia Lehrer, and I'm the Executive Director of the Woonasquatucket River Watershed Council. And I am so excited to share with you the work of some wonderful students from Smith College in a capstone program that they did on data analysis, which you will hear a lot more about, where these brave students took on all of the data that has been collected over the decades on the Woonasquatucket River Watershed, and they have analyzed the data and made some distinctions about what's been happening in the watershed over the years. So without further ado, I would love to introduce Katie, Quinn, Janaya, and Nina, who are our capstone team, and they will tell us all about their data analysis project.
Hi, everyone. My name is Quinn. I'm a statistics and data science and biology double major, and my plans after graduation are to move back home to Central Coast, California, and apply to some biostatistics or data analytics fields.
Hi, everyone. My name is Katie Cummings. I am also a senior graduating in May. I'm double majoring in statistics and data science and psychology. Post-Smith, I am moving to Philadelphia as I've accepted a position to teach third grade special ed with Teach for America.
Hello, everyone. My name is Janaya, and I also study statistics and data science along with Spanish. And post-grad, I plan on moving to Boston.
Hi everyone, my name is Nina. I am also a statistics major and I'm double majoring in biochemistry. And after I graduate this year, I'm going to be doing a postbac at Brown University in Rhode Island.
Okay, so this is just a little bit about the class that we're taking, what is its goals, and we're actually going to have our professor Shia elaborate a little bit more about that.
Hi, sorry, I was muted. Hello, everyone. Thank you so much for the opportunity. Yeah, I just want to introduce you to our SDS Capstone course. So as you can see on the slides, the students put. So the SDS Capstone course, we have that every semester in our statistics and data science department at Smith College. I probably first should say I'm an assistant professor in statistics and data science department at Smith College. And so this capstone course is modeled as a simulated data science consultancy. So students will work on a real world data problem a sponsor provide, like Alicia did this semester. And the students who take the capstone course are all Smith College senior undergraduate students majoring in statistics and data science, like Katie, Kun, Janaya, and Nina. As you can see on the slides, so students work in teams of 3 to 5 on a project. And these students have foundational training in statistical modeling, computing, data visualization, data wrangling, and data ethics, etc. Some students, like some of our students right now, they also double major in psychology, in biology, in biochemistry, and just many other majors. So they may also have domain knowledge in broad areas. We are having this class every semester, so we keep recruiting sponsors. So we are also recruiting sponsors for next year and for the fourth semester specifically. So an ideal project is a project that has enough protein to mastering our data science curriculum. Meaning that this project should require students to use major programming languages such as R or Python and work on non-trivial real-world data. And give you some examples of past successful projects. They may involve building statistical models, creating data visualization, web scraping, developing a web or mobile app, or developing a database, etc. It's really just what you want. So then students will do the consultants for you. So the end result could be a research paper, a statistical model, a web or mobile application, a dashboard, or whatever else seems appropriate. Yeah, so this is just the basic what this course is about and what kind of projects we are looking for. And I actually don't know if I can send the information of our professor who is going to teach Capstone next semester. And if you have projects you want students to help with, and then you can email him. But I can see here, I think I can only send the chat to host and panelists. I'm not sure if everyone.
See, what we can do is if people are interested and think they might have a great data project, just put your name and your contact info in the chat, and we will pass that along to Shia and her colleagues.
Okay, that sounds great. I'll do that. Thank you so much.
Perfect.
So students can go ahead to do their presentation. Yeah.
Thanks, Shia.
Thank you.
Oh, sorry, tap it first. That's annoying. Yep.
Thank you so much for that introduction. So let's get started on what we've been working on this semester. As we know, the Woonasquatucket Watershed is a vital resource and plays a pivotal role in the environmental health of the region of Rhode Island, providing water for various uses and serving as a haven for diverse wildlife. But despite its importance, a detailed understanding of the water quality trends within this watershed has been elusive due to gaps in comprehensive exploratory data analysis and challenges in consistent monitoring. So our research is significant for several reasons. Firstly, it affects the lives of many, with more than 25% of the state's population depending on this watershed. Secondly, it addresses both immediate and long-term environmental needs essential for sustaining biodiversity and ecological balance. And lastly, the social impact of and economic aspects cannot be overlooked, as the watershed enhances community well-being and local economies through recreational activities and serves as a vital water source.
Wait. Oh, okay. There we go.
So our study utilizes an extensive data set covering 28 sites along the watershed from 1990 to 2021, which is over 30 years of data. So we utilized the programming language R and various packages for statistical analysis, focusing on 7 key water parameters that we narrowed down with Alicia. And our methodology included rigorous data pre-processing to ensure the accuracy and reliability of our findings, which included cleaning data, handling missing values, standardizing measurements, and standardized measurements across different collection points.
So our data, as you know, looks at a total of 28 watershed sites across Rhode Island. And to make our project a little bit more specific, Alicia provided us with 7 parameters that we looked at for these watershed sites, along with the years and decades. With doing this, Alicia also asked us to make a way to visualize the watershed locations. So we created an interactive map. There's 2 different types, one that can be made available on a website and the other one that is for data analysis. I'm gonna go and show you this map really fast. So using R, we created an interactive map that you can change how you view the watershed locations. This is my personal preference because you can see where the watersheds are more clearly with like water. And you can click on various sites and they will provide all of the data that is about that site, which is useful when you're determining what is upstream and downstream. And this is just more of an example of what you see when you click on a data point. So in doing this, we also ordered the upstream downstream effect from descending order by the site's latitude. And these are tables which show where the sites are. So as you can see, the highest one is site number 52. And the most downstream site is site number 227. And this is for all 28 sites.
In terms of our parameters of interest, we decided to go with the 7 most important ones that pretty much determine water quality. I'll go through them very quickly. So they are known as the chloride, the phosphorus concentrations. We also looked at dissolved oxygen concentration, fecal coliform, nitrogen, enterococci, and temperature. All of these parameters are used when assessing for water quality due to their impacts on not only aquatic life, but also public health.
Okay, so we'll begin with some summary statistics about our parameters overall. This table shows, regardless of decade, year, or site, what are the mean, median concentrations of each parameter and what kind of sample sizes are we looking at. So you can see that the sample sizes are pretty variable, and that will impact the reliability and the predictive power of the models that we've made. One thing to note is that the enterococci and fecal coliform are both bacterial parameters, And so they are in log form, but if you were to take them out and get just those raw numbers, then they will be the ones that you see below. It should also be noted that fecal coliform was collected about half in MPN per 100, and that is most probable number of colonies, I believe, per 100, and the other half is in CFU per 100 milliliters. But we did a little bit of research, and although there isn't a conversion factor, they're effectively the same. So we just included both of those in our calculations. So this first plot here that we have is the density plots of each parameter. And so we have density or log density on the y-axis and concentration on the x. And so you can see that this provides a good sense of the spread and the normality of the data. You see that fecal coliform and enterococci have the most normal looking distribution. Enterococci might be a little bit bimodal, so there could be reasons for that. That future work would help us realize. But yeah, these are where most of the points fall in the data. And next we have our bar plots, and this is a plot of mean concentration of each parameter over the years. So this gives us an idea of what kind of trends we're going to see with the data, and you'll notice that some are missing little gaps or some have less bars than others, and that's just because at any point in our data processing process, these data could have been eliminated because there were weird looking outliers, there were NA values that are affecting our summary statistics, and so there wasn't always sufficient valid data for every single year and every single site, so you may see a couple of gaps. And this is more bar plots, but this is showing the concentration of each parameter over the sites. And the sites in these graphs are all ordered in ascending order. So we're starting with site 227 at the lowest latitude, the lowest latitude, and working up to WW061, and that's the ID for the most upstream site. Next, we have this facet wrapped graph of each parameter and its concentration over the decades. So what I did was I grouped all the years together, and we're looking at 4 decades in total. Of course, in the 2020s, we only have 2 years for that. So that data is not quite as reliable. We'll have a better understanding of that, in 8 years. But yeah, so we have the 90s, which is nice. We have all 3 full decades prior to that. And yeah, this is how it's been changing over time. And as far as statistical analysis, what we did was like an analysis of variance type of test called the Kruskal-Wallis test. And we used it because it is more appropriate for data that has multiple groups. Obviously our groups for year 32 and our groups for site were 28. So we have a lot of really fine-tuned comparisons that we're looking at. These p-values, they all say 0 and they all are effectively 0, which means there's a very small chance that these changes are just due to chance and not because of a cause. And then we determine that significance by comparing the p-value to our alpha number, which is 0.05, and that's standard to use. So this is the same thing, but now we're looking at the difference in we're looking at the change in each parameter across different sites instead. So what this tells us is that there is significant changes, not due to chance, in all of our parameters between the different sites. So now we have some linear relationship models. This one is for the mean concentration of each parameter over the years. And you can see that pretty much all of them are looking like a positive correlation. This can't exactly be used to make any causal claims because we aren't using only numeric variables, but I just added this just so we could get a nice visualization of where the directionality is going. Let's see. Yeah, so this is the next one. This is by site. When making the data frames that were just having to deal with site, we had significantly less data as opposed to year. And there was not enough data nor strong enough of a correlation for a regression line to show up on these plots. So there isn't really directionality in these changes, nor is there a whole lot of relationship between the concentrations and the sites. So Alicia also asked us to make plots that were tracking the changes in all parameters over time, but in a single site. So this is site 308. And so anywhere that you kind of see a peak, that's somewhere where, oh, look, 2014, that's kind of higher than the rest of them, what's going on there. And then, you know, the Watershed Council will look at that and maybe say, oh, something happened in 2014 that would explain this. Again, anywhere that you see less bars or no data, that was just, we didn't have the data. Our next one is site 227. That's the most downstream of all of the sites. So this is interesting to look at. Yeah, again, not a whole lot of change in these specific sites, but still interesting. Site 437, same deal. And same one for site 635. 635 and 437 were both kind of in the middle. But yeah, these sites are all what is being currently monitored. So in the future, we'll have a lot more data to work with for those.
Yeah, so with these findings, we saw that there are significant correlations between year and changes in the water quality parameters such as chloride and fecal coliform, excuse me. And as Quinn said, since that we are looking at non-numeric, we can't necessarily say directional correlations, but we can see that there has been changes. And with the high R² values for chloride and fecal coliform, these suggest that the parameters are more predictable over time, which is crucial to understanding more of the public health risks that are associated with water quality. Additionally, we have lower R² values for parameters like dissolved oxygen and phosphorus. These highlight the complexity of ecological dynamics of the watershed, suggesting that current management strategies may need to be reevaluated to address these variables more effectively. There are standardization issues and overall, it's overall hard to draw from a broad range of exploration. Most of our time was spent during the cleaning and organizational processes of the data, considering that there wasn't necessarily consistency in the data collection. So the analysis is a little bit more vague at the moment, but that is okay because we hope that the watershed will continue to look at this and see what the data we've offered shows for each site. Additionally, we want to note that there are shifts in the EPA standards for measuring some of the parameters, most notably is fecal coliform, as fecal contamination highlights the need for some updated and consistent methods in monitoring water quality to align with public health guidelines. In 2012, the EPA changed its standards and said that for public health risks, the bacterial of E. coli is needing to be analyzed and collected to see whether or not it is healthy for people for recreational uses.
In terms of the EPA standards that we found for the state of Rhode Island, we believe that the majority of the parameters, such as phosphorus, dissolved oxygen, and chloride all fall within the maximum, which means that the water quality has progressed over time is getting better. For example, when we looked at the most downstream site, we found out that the average phosphorus concentration was 0.25 milligrams per liter, which is just at the rate in which it should be according to EPA standards. Similar with that, with the chloride concentration, the average for across all sites was 43,000 micrograms per liter, which the highest concentration being 90,000. And according to the EPA standards, that should be around 2,300,000 to 860,000, which would definitely mean that there would be a chronic or acute problem. So looking at in terms of those parameters, the water quality within those sites are doing really good. That being said, moving forward in future directions, due to time constraints, we were unable to do so. But if another person were to take on this project, we would want to explore more non-linearized models, such as logistic or exponential regressions, to be able to fit the relationships between year and water quality, since our R-squared values were so low. We would also want to investigate the effects of different sites within the watershed to understand spatial variation, meaning that we would want to look into external factors that don't necessarily exist within the watershed, but might be due to human interaction within Rhode Island. And as what Katie had mentioned before, transitioning from enterococci to E. coli recommended by the EPA standards, this would allow for a more effective assessment of water quality that, of course, is within accordance by the state. And lastly, in order to improve data collection methods and to reduce inconsistencies, we would most definitely recommend taking measurements at the same time every month, every time per season, which would allow for less variability, less bias, and just an overall better understanding of the data. So that being said, in conclusions, our work was most definitely preliminary, but we were able to provide a foundation to be able to understand the data set and the values within correlation within the Woonasquatucket River Watershed. And we, moving forward, urge environmental scientists and policymakers to use our findings to better assess the water quality, as well as to make other future directions.
And of course, a special thank you to our sponsor, Alicia, and all of those people over at the Woonasquatucket River Watershed. It's been great working with you guys, and our Professor Shia and our TA Mimi.
Thank you.
Thank you so much, Katie, Quinn, Janaya, and Nina. We really appreciate your presentation. Now, if anybody has questions, we have a little bit of time left where we can, if you have a question, you can put it in the question and answer. Chris, do we have the ability for people to actually speak, or do they have to just type to get to us?
It would be speaking. I'm sorry, just typing at this point.
Just typing. Okay. So it looks like we do have a question here, and it is the GIS mapping with sample data available as shown in the presentation?
Yes, so there are two things that are available. There's one, just like I'll show you actually. Sorry, one second. Okay, let's see if this works. Yay. Okay, so this one is available. Just by like linking it, you could web link it. This is not as like, it doesn't give all the data as much, but it does like locate the watersheds. As for like the other one, this one, this is available. It is coded. There's an HTML link that can be provided. Yeah, so you can look at it. Yeah.
Can you possibly put those links right there to those in the chat?
So I think that what I can do is I can send you afterwards I can definitely send I can send it afterwards because I don't know how well the code will run through the chat, but it can definitely be provided very easily at the end of the webinar.
Perfect. Okay, it looks like we have another question. Sorry, Chris, did you want to say something?
Oh, yep, I was just about to announce Ann Johnston's question. So if you can take it from here, Alicia.
Okay, so Anne's question is, does anyone know the source of the E. coli? Is it from a sewage wastewater treatment plant? And that is such a good question, Anne. And the answer is, it could be. If I had to take my very best guess where that E. coli is coming from, it's probably coming from stormwater runoff and from illegal sewage tie-ins to the watershed. You just can't get away from that at this point, unfortunately. I do think that our cities and towns along the Woonasquatucket are doing their best to track down and eliminate those sources of discharge. But I have seen in some cases that just geese can spike that E. coli number. Also, places where people aren't picking up dog poop, we can get a huge amount of spike in there. But I think ubiquitously, I've been seeing that there have been inputs from illegal tie-ins to storm drains. So that's my best guess. We don't currently have the capability to test the DNA sources of that E. coli, so it could be bacteria from any warm-blooded animal. So it's really hard to say. All right, so Bridget had oh, go ahead, Chris.
Yep, and we did update the settings, so if folks would like to speak, I can turn that option on. I see Cynthia Mulvey does have her hand raised. So if you want to address the questions that just came in, and then I can let Cynthia speak as well.
Great. Okay, so we've got another question from Bridget that says, what documents reporting will be provided to WRWC besides GIS, sample data, mapping? What other things are you guys sending WRWC? And I know it's a lot, so go for it.
So I have written up and sent to Alicia just a couple days ago, actually, a sort of like workflow for how we approach this problem and just some tips for future data collection. So yeah, that document, it just kind of says, okay, you guys gave it to us in an Excel workbook. So we then went into that Excel workbook and we did a little bit of data cleaning in there, but I primarily prefer to do it in R. So we got rid of a couple columns that we didn't need. And then what I did was put that into R, uploaded it, changed some names, moved stuff around, added columns, got rid of missing values, all that kind of stuff. And I then kind of wrote a skeleton for how you would approach that in specifically in R, but it could apply to other programming languages as well.
Yeah, and in addition to this, for our course, we have to write a term paper, which is basically kind of somewhat similar to what we just went over, but not at the same time. And that will also be provided to the watershed, so you can have more of a detailed understanding. And that includes all the graphs, that includes tables, that includes all additional things. We also have additional code that includes visualizations and some tracking that we didn't include in our term paper for space. And all of that code will be also provided to the watershed.
Thank you. That was a very comprehensive answer. Totally appreciate that. And I just want you guys to know that I worked with these students every week and they did so much research on this project and did their very best to make the data meaningful. And I just so appreciate that about you guys. I'm going to take a question from Kevin, and then we'll go to Cynthia. So Kevin says, would you guys agree with the statement that water quality does not appear to be getting either better or worse? Neither better nor worse.
I believe we all would agree with that because in terms of individual parameters, we do see an improvement in dissolved oxygen as well as chloride. However, in terms of others such as phosphorus and enterococci, those terms, it doesn't necessarily seem to be increasing or decreasing, but staying at a threshold that in terms of the EPA standards is, I wouldn't say not okay, but at the maximum. So what I had said previously, for example, of the concentration of phosphorus, the EPA standards say that the maximum concentration within a certain body of water should be 0.025 milligrams per liter. And unfortunately, we looked at the most downstream site for that, as well as the averages across, I believe since the beginning of this data collection to currently now, which I believe the last data point was 2021, it still seems to be staying at that average. So we would most definitely agree that some aspects are getting better. However, some aspects don't seem to be necessarily getting worse, just not changing.
Yeah, a big part of this project was that, as Alicia mentioned, we're the first ones that have touched this data. And so the point of this report is to be an exploratory preliminary analysis that can be used as a framework to then do other work on and make causal claims. But our study, we cannot make any causal claims about this because the whole, correlation causation thing, this also has to do with an entirely another field of, biology and ecology that we're not particularly well versed in. And another thing, we didn't get the chance, due to time constraints, to analyze everything that we wanted to. I did my Kruskal-Wallis test, but I haven't been able to find time to do any post hoc tests that would control for experiment-wise error rates. And so yeah, this is just giving us an idea of where we're at now. And then once we straighten out that data collection process and do more analysis in the future, then we may be able to make some more solid claims about the water health.
And we do really recommend that, and we recommend like the use of like having a someone who does data science or is able to do something in our in order to continue the purpose of this research project.
Yeah, and we did bring that up in-house with the students, thinking about, well, now what do we do? And luckily they did provide us with the methodology that they use to analyze the data. So we foresee from our perspective having an intern every year just keep updating the data and maybe doing some additional analysis. If you guys could go to your slide that shows site 227 for just 1 second. Like more toward the end there. Okay, right back there. So I'm pretty sure this site is in Denigian Park. And one of the things that I was super interested in, it looks like we've got enterococci and fecal coliform is only going through 2010. And I'm guessing that's because Watershed Watch stopped analyzing for that parameter at that point and switched over to enterococci at that point. And, I was hoping that I was going to see a significant decline after 2015. But as you can see here, you can't really see that decline. I do know that we have some friends here from the Narragansett Bay Commission that did the whole sewer interceptor project, taking offline all those combined sewer overflows. There were 19 of them going into the Woonasquatucket. And I was hoping that we would see like more of a change over time, but we haven't seen that yet. So maybe we're not analyzing the right way, but it was just something that I was hoping that we would see. We still have time for Cynthia to ask her question. Cynthia, you want to pop on for a sec? Okay, Cynthia looks like she typed it into the chat. So she would like to see more recent analysis of Stump Pond and the Stillwater Inlet, since these spots weren't tested from 2014 until 2022 and last year with URI. And Cynthia, I believe you're the one who is doing the monitoring at these sites. Is that, I think I'm right about that. In comparison with data you have in prior years. I don't know, team, do you have any response on Stump Pond and Stillwater Inlet? We'd have to, I mean, no, they would have to know the site numbers for
Yeah.
that one.
We don't know the names of the sites. If we knew the site, I don't. Yeah, I don't know what sites those are. My response would be using the code that will provide. That's totally manageable. You would just have to like swap out, for like example, for this one, you would just have to swap out the sites. And then, you know, rerun the code and you would be able to get visualizations for those sites. And you could do that in comparison to other years. Yeah, it definitely is manageable, but I don't know if we have those.
Thank you. So Cynthia, I'm gonna just say, like, you know, contact me afterwards and I can help you with the methodology and the re-sorted and clean data that they gave back to us to look at those questions. Any other questions? Okay. All right, I see a question from Sam, or he says, thank you, good job cleaning all that data. Sam knows what our data looks like. And acknowledging the constraints within the data set, did any one of the variables stand out to you showing a stronger trend? I'm a little curious about phosphorus.
Well, let's go back to those.
Yeah, let's look at them.
Yeah.
I mean, we could hear this one.
Yeah, chloride and fecal coliform have the highest studies.
If we go back to the ones with the regression lines.
Okay, we can do that. Is that before? This one?
There she is.
Okay.
Yeah, so nothing has a particularly stronger looking correlation than anything else, except, you know, some are looking like there's little to no correlation. Yeah, it looks like chloride, enterocoxi, and what's that, nitrogen? Those all look like the strongest positive correlation that we have. And that's per year. And if you go to the next slide for site, yeah, nothing super stands out. No, there were very few extreme outliers that I had to get rid of. But all of this looks pretty. But yeah, as you can see, all of them look pretty consistent on average.
And I'll just add something really quick. When we looked at the R-squared values, which R-squared is a measurement to test whether how strong the model can predict what's going on, and that ranges from 0 to 1. However, when we looked at it, chloride and I believe it was fecal coliform, they have the highest R-squared values, and those values I believe are around like 0.5. So in terms of like statistical analysis, that's not necessarily the strongest. Yeah. So
yeah, so really not convincing one way or the other.
I do think that they're like, personally, I'm interested in like how like more public health based. So it was definitely interesting seeing like looking at the variables of fecal coliform and enterococci. Yeah.
Thank you. We've got a couple more questions here. Bridget had another one. She said, research prompts questions as to when sampling is taken, dry weather and no rain for 72 hours, or wet weather, and how rain events have increased over time affects the parameters. And Elizabeth had a similar question. Elizabeth also, as we know, is the director of URI Watershed Watch. So, you know, very, very similar question. Could comparisons of wet and dry years be done with the data? That's the question to ask.
Yes, there could. We would just have to have that.
Whatever year.
I could supplement that into the data set that we already have, where we could kind of overlay those and, you know, look at them transposed, that kind of thing. I don't think that was present in the Excel workbook that we got, but if we had it, the short answer is yes.
It's a good point. So, you know, we probably could pull the weather data and the rainfall data from each of the years and do the comparison in that way. So I think that makes a lot of sense, Elizabeth. I'm curious about that as well. And maybe if I do another Smith Capstone project, we'll ask some of these specific questions that you guys are raising here. We have a couple more minutes, so if there's any other questions, feel free to put it in the chat. We'll just give you a minute here.
Alicia, if any questions come up after the webinar, is there a particular contact somebody could send their questions to?
You know, they can just send them through me. I think everybody on this webinar knows how to find me, but Chris, if you want to just put my email into the chat for the attendees today, that's probably the best way. And then I can reach out to the students or see what I can tell you.
There's this is all of the emails for us included.
Nice.
Yes.
Elizabeth also says that we have the Kingston station data, which I'm not sure what that is, but perhaps TF Green Airport. Oh, I see, for weather. But TF Green Airport or another station would be better for the analysis. And we've got a lot of thank yous coming in here. So I think that concludes our Lunch and Learn webinar for the day. And I just really want to take a minute to thank these students, Katie, Quinn, Janaya, Nina. You guys did so much work on this project. I'm so impressed with how you delved into the research for this project, including analyzing that ridiculous amount of data that we sent you. And I know that you're all going to do so amazingly well in the next phase of your lives. And Shia, again, thank you for allowing us the opportunity to have a data analysis project with your awesome students.
Yes, it was a great working with you all.
Yeah,
we really enjoyed it. We really have had a lot of fun. Yeah.
All right,
Thank
everybody.
you so much, Alicia.
Again, remember, if you have a data analysis problem that you would like to solve, don't forget to send that info to Shia or to Ben, or if you want to just send it straight to me, I can pass it along. And everybody have a great rest of your day.
Thank you.
Thank you, everyone.